So welcome to the first tutorials on Krok 1 Physiology Base, Krok 1 Physiology Base. And this is one of five videos to come. All right, or 5 tutorials to come. So let's begin. Let's begin. A patient with hypersecretion of the gastric juice was recommended to exclude from the diet concentrated bolus and vegetable decoction because of their stimulation of the gastric secretion. What is the dominating mechanism of stimulation of secretion in this case? So they are talking about hypersecretion of the gastric juice. Hypersecretion of the gastric juice. So here we are referring to what? Gastrin. So gastrin is simply the hormone that stimulates the secretion of the gastric juice. It stimulates the secretion of the gastric juice. So this is what? Gastrin. And of course, the gastrin affects some cells. These are called what? The parietal cells. Parietal cells of the stomach. Parietal cells of the stomach. And they release what we call gene cells. They release what we call gene cells. So basically over here, we are talking about stimulation of the gastrin production. Gastrin production by the gene cells. Because this is what? Stomach. So we're referring to C. All right. Person felt thirsty after staying in the heat for a long time. Signals of what receptors caused it, first of all. You know, when you stay in heat for a very long time, what happens is that you begin to feel what? Thirsty, because fluid gets lost out of your body. And this phenomenon, or the receptors that are able to identify these things, we call them osmoreceptors. So they detect the osmolarity of the body. They determine what? The osmolarity. And these osmoreceptors, they are found in the hypothalamus. So over here, we are talking about what? Osmoreceptors of the hypothalamus. So when there is too much concentration of salt in your body, it begins to secrete, or it stimulates the hypothalamus to secrete or to make you feel what? Thirsty, so that you will drink in what? Water to nullify or to neutralize the osmolarity. And the reverse is also what? True. In case of when there's no too much salt over there to stimulate, to quench your thirst and all of these things. So basically, we are referring to what? Osmoreceptors. So they determine, or they detect the changes in osmotic pressure. And they contribute to osmoregulation, controlling fluid balance in the body. They control fluid balance in the body. Fluid balance. So here you are thinking of A, osmoreceptors. An individual is characterized by a round face, broad forehead, a moon-like type of Mongolian type of eyelid fold, flattened nasal bridges, permanently open mouth, projecting lower lip, protruding tongue, short neck, flat hands, blah, 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 blah. What are you thinking about? Definitely this person is suffering from what we call what? Down syndrome. These symptoms are typical for Down syndrome. You know, Down syndrome has to do with what? Trisomy 21. The last time we discussed about the different types of syndromes, Klinefelter, Down syndrome. Turner syndrome, you already know them. In Down syndrome, we are talking about trisomy 21. Trisomy 21. And every single bit of the features here is actually describing trisomy 21 or Down syndrome. Down syndrome. In Klinefelter, we have what? XXY, XXY, XXY. In Turner, like I told you, they've turned down, so XO. Or they didn't turn up. So the X chromosome didn't turn up. So you have XO. But this is what? Down syndrome, trisomy 21. Trisomy 21. So the answer is B. PRulent endometritis. PRulent endometritis. Developed in a woman after delivery. Treating with antibiotics inhibitors of Morin synthesis was ineffective. Wide spectrum bactericidal antibiotic was administered to her. 6 hours, there was temperature and shivering. Muscle pain has appeared. Blood pressure has dropped. There's oliguria. What is the main reason for the development of this condition? What is the main reason? So first of all, they've told you that this person is suffering from what? Purulent endometritis, and antibiotic wasn't working. Antibiotic is not working. And there is what? Now, after 6 hours, even after giving a blood spectrum antibiotics, there's what? There is still temperature and there is shock. So this is tell you that this person is going through what we call septic shock. Septic shock. That means shock that arises as a result of what? Severe infection. It arises as a result of severe infection. And another name for septic shock, we can also refer to it as what? Endotoxic shock. Endotoxic shock. So we are referring to severe infection leading to shock. So your answer is endotoxic shock. So E should be your answer. We have secretion of which gastrointestinal hormones is primarily decreased in the patient with removed duodenum. So there's a removed duodenum. So the question now is, what type of hormones or enzymes are produced by the duodenum? What type of hormones are produced by the duodenum? And the duodenum produces what we call cholecystokinin and secretin. Cholecystokinin and secretin. I repeat, cholecystokinin and then secretin. So here we are thinking of what? Of E. Because if that place is removed, then their hormones will be reduced, or the enzymes present at that area will be reduced. And that's why over here, there is what? A decrease in the gastrointestinal hormones due to the removal of the duodenum. Of the duodenum. So here we think of E. We think of E. Of course, if they talk about the gastric part, of course, you could be thinking of what? Gastrin. Gastrin. All right. A 2-year-old child experienced convulsion because of lower calcium ions in the blood. What structure is decreased? Or which organ is having a problem? Calcium is produced by what? Of course, parathyroid gland. It is produced by the parathyroid gland. So here we are looking at what? A. So if A is impeded or is faulty, what it means is that calcium could be very low. Calcium could be very low. All right. And you know, when calcium is low, it can cause what? Rickets. Could be what? Vitamin D deficiency. Vitamin D deficiency. And apart from that, is what we're having here. convulsion, and every other thing, numbness, arrhythmia, tetany, all of those things can ensue as a result of low calcium levels. So your answer here is A. While emotional excitement, the heart rate in a 30-year-old person was 112. What part of the conducting system? We said if it is between 60 to 80, we are looking at what? Sinoarterial node. 60 to 80 is what? SA node. 59 and then 40, we are looking at what? Atrioventricular node. And then let's say 20 to 39, we are talking about what? Bundle of what? His. Then less than 20, we are talking about what? About the Purkinje. We are talking about the Purkinje fibers. So over here, because the person is emotionally excited, of course, it could be more than what? The 80. It could be more than the 80. So over here, we are looking at what? Sinoarterial node. Sinoarterial node. So during exercise, of course, the SA node will beat higher than usual. Higher than usual. So your answer is A. Usage of contraceptive with sex hormones inhibit the secretion of hypophysial hormones. Again, you are using what? Contraceptive. And you know contraceptives are used to control what? Birth. They are used to control birth. So they want to know, what does this hormone actually do in order to prevent a child from, or to prevent uh, fetus formation. So there's a secretion of which indicated hormone. So the question is, what will be affected? What will be affected? So basically, what contraceptive they do is that they inhibit the release of what we call luteinizing hormone. They release what we call what? Luteinizing hormone. And when this luteinizing releasing hormone are inhibited, they will suppress the level of follicle stimulating hormone and then the luteinizing hormone. So again, we are looking at what? Follicle stimulating hormone and then luteinizing what? Hormone. So these are the major, let me say hormones that will be inhibited when you use contraceptive. When you use contraceptives. And I believe the ladies can know this better for us. So the answer is what? Follicle stimulating hormone. Follicle stimulating hormone. It will be affected. So your answer is A. During the breakdown of acute respiratory infection in order to diagnose influenza, the express diagnosis, to diagnose influenza, the express diagnosis, they based on revealing specific viral antigen in the material is carried out. What reaction is used for this? So what they're trying to tell us that we are having a viral infection. So among these, which method is used to identify a specific viral antigen, a specific viral antigen? Unfortunately, you are not a lab technician, so maybe you might find it difficult to understand. However, we are talking about what? Immunofluorescence. We are referring to what? Immunofluorescence. Immunofluorescence. And it is a lab technique which is based on the use of specific antibodies which have been chemically conjugated to fluorescent dyes. It is the use of specific antibodies which have been chemically conjugated to fluorescent dyes. To fluorescent dyes. And now these antibodies bind directly or indirectly to cellular antigens. They bind directly or indirectly to cellular or cell antigens, and they provide a very rapid and specific diagnosis. They provide a very rapid and specific specific diagnosis for viruses. And that's over here. We'll go in for what? Immunofluorescence. Immunofluorescence. Because the question here is asking for what? Specific viral antigen. So your clue here is what? Specific viral antigen. And we'll use what? Immunofluorescence. So your answer is D. 38-year-old woman was admitted to the admission diagnostic department with bleeding, urethral bleeding. What are the most likely changes of blood? So there is bleeding. There is what? Bleeding. And sometimes when there's blood loss, what it means that there will be what? There will be reduced, because there's blood loss, of course, there will be reduced volume of what? Of blood in the body. Reduced volume of blood in the body. And the ratio of, especially, we're also talking about especially the red blood cells, especially the red blood cells. They go in out of the body a whole lot of what? Times. So the ratio of the volume of the red blood cells to the total volume of blood will be decreased. Will be decreased. Will be decreased. So here we are looking at for reduction of hematocrit rate. And hematocrit is simply a ratio comparing the volume of red blood cells to the entire blood volume. The volume of the red blood cells to the entire what? Volume. And this person has been what? Is bleeding. You know, in bleeding too, we can see a lot of the red blood cells also what? Going out. A lot of red blood cells going out. But no doubt, they can also be what? Leukocytosis or redistribution of blood in the body. Okay, no doubt about that. But first of all, there'll be what? A reduce in hematocrit rate. Reduced in hematocrit rate. So here we are looking at what? At D. At D. At D. All right. There can be, but we'll go in for what? The hematocrit rate. Because from the hematocrit rate, you will know that, oh, this person is really suffering from RBCs. Is really, really suffering from what? RBCs. The RBCs are low. So let's make sure we save this guy as soon as possible. Because the RBCs contains what? The hem, which must bind with oxygen to perfuse the body. Great. Now, due to the action of electric current on excitable cell, there appeared depolarization. Depolarization. So what ions are we looking at? For depolarization, we talked about it several times. We've not talked about it, but we talk about the T wave or something like that. But when there is depolarization, it means there is an influx of sodium. There is an influx of sodium. There's an influx of sodium. Influx of sodium. Influx of sodium. So here we are thinking of what? Of D. Of D. Of D. There's actually what we call action potential. In action potential, we have the depolarization state, whereby there is what? Or the membrane becomes permeable to sodium. It becomes permeable to sodium. And so therefore, the membrane becomes what? Positively charged. It becomes positively charged. It becomes positively charged. And these sodium channels play a very big role in the initiation and conduction of action potential, thereby increasing the conductivity or the nerve conductivity. So here we're talking of what? Of the sodium channels for depolarization. Now in, how do you call it? Hyperpolarization. What do you think will be? Of course, potassium. The opposite will be true, potassium. But here we are referring to what? Depolarization. So sodium. the high level of lactate dehydrogenase, LDH, showed an increase. Do you remember LDH1 and LDH2? Of course. You remember in biochemistry and even in pharmacology, we did this. And we said anytime inside the body there's increase of LDH1, LDH2, and even we mentioned what we called creatine kinase, creatine phosphokinase, creatine phosphokinase. And we said LDH1 are mainly found in the heart and also sometimes in the RBCs, And LDH2 is also found, or it can also be found also in the heart or reticular endothelial cells or the red blood cells. You can find them there. So over here, we are definitely looking at the heart as a problem. We've done this before, and I will not waste or spend much time on it. We've done this before. So over here, we are looking at for myocardial infarction. In some books, they will add the creatine kinase to it. Creatine kinase. That's CK. Or creatine phosphokinase. We've done it. We've done this already. So please do well to revise. And you see, we keep repeating them. There is only one hormone among the neurohormones which refers to the derivative of amino acid according to classification. Derivative of an amino acid according to classification. So all that they're trying to tell us is that which one of these has a link with amino acid. And here, what are we looking at for? First of all, so the amino acid derived hormones are derived from thyroxine and tryptophan. And they include epinephrine, non-epinephrine, and then non-epinephrine. So please take note. So these are some of the classifications of the amino acid derivatives. So we can talk about tyrosine, tryptophan, all of these are all linked with it. Then again, we can be thinking of how do you call it? Since we mentioned tyrosine, we know that tyrosine are produced by the thyroid gland. Thyroxine, or I'm saying that we can also have what we call tyrosine, or T334. They are all amino acid or derivative. T334, they are all amino acid derivative. And these are produced by the thyroid gland. Then we can also talk of melanin or melatonin. All of these are produced or are derivative of amino acid. So again, what are the derivative of amino acid? We can think of tyrosine. That is TYROSINE. Thyroxine, then tryptophan, TRYPT, tryptophan, then PHAN, tryptophan. So we have thyroxine and tryptophan. These are derivative of what? Of an amino acid. We can also talk about thyroxine. We can talk about thyroxine, and we can talk about what? About melatonin. So, and I'm saying all of these things are all derivative of amino acid. So here, what are we looking for? We're definitely looking for what? For melatonin. So your answer is A, melatonin. However, if any one of these was also present, we could have also gone in for it. That's why I've given you all of them, so you know when you see them. You will know it when you see them. We have a sterile petri dishes and pipettes are necessary to prepare for microbiological tests in a bacteriological laboratory. What way of sterilization should be applied? What way of sterilization should be applied? Now we're talking about sterile petri dishes and pipettes. Dishes and pipette. How are you going to sterilize these equipment or these, basically equipment? So basically, we are going to sterilize them in a high or at high temperatures. We are going to sterilize them at high temperatures, because at high temperatures, bacteria or microorganisms, they all die. So that's what sterilizing them at high temperatures, And this phenomenon is called dry heat sterilization. Dry heat sterilization. And some of the things that we use dry heat sterilization for include the following. So if I were you, I'll write them down. One of the things that we use dry heat sterilization for, they include one, metal instruments. Metal, metal, metal instruments. We can talk of glass wire, glass, GLASS, glassware, AWARE. Then think of syringes. Sterile petri dishes. Pipettes. Paper wrapped items. Sometimes you can even talk about fats and even powder. You can talk about fats and powder. Okay. So that is what dry heat sterilization, some of the instruments or some of the things that we can put them under what? Dry heat. Then we also have what to call pasteurization. So in this question, we're talking about dry heat. Okay. But I want to add some of other things in case question is twisted. All right. In case question is twisted. So we also have what we call pasteurization. This is a process in which water and certain packed and non-packed foods are treated with mild heat. So we're talking about what? Either packed or unpacked food that has to be what? Undergoes a mild what? Heating. And usually the heating is less than 100 degrees Celsius. And this will also what? Eliminate the pathogens. It will also eliminate pathogens. So that's what we call what? Pasteurization. So sometimes even in our microwave, where we heat food and stuff like that, we can technically describe it as what? Pasteurization process. We can. Okay, they also have what to call tenderization. So this is also what? A process for sterilizing substances, of course, food, that are used to kill heat-resistant endospores. Heat-resistant. When I say they are heat-resistant, they are not afraid of what? Of heat. They are not afraid of heat. So we use what we call tenderization. All right. So basically, these are some of the things I wanted us to talk about. So basically, we're talking about dry heat sterilization. So here, we can go in for A. All right. Let's say a patient after hypertension stroke does not have voluntary movement in his right arm and leg with increased muscle tone. increase muscle tone in these extremities, what type of dysfunction of nervous system is it? Guys, do you remember when we talk about increase in tone? When we talk about the different types of paralysis, about peripheral and central, we said in peripheral, sometimes it leads to what? Flaccid kind of what? Paralysis. Flaccid kind of what? Paralysis. And then in central, would normally lead to what? It leads to increase in muscle tone. Leads to increase in muscle tone. Increase in muscle tone or spasticity. Spasticity. So over here, what are we looking at for? We're looking at what? Central paralysis. Central paralysis. Please, I've talked about the different types of paralysis, peripheral and central. Please, if you've not watched the video, do well to watch the video again. Or if you've forgotten, try and then revise it again. Okay. All right. And again, we said under peripheral and central, we said in peripheral, it has to do with lower motor neuron. Lower motor neurons are affected. In central, upper motor neurons are affected. Upper motor neurons are affected. All right. Blood analysis of a patient showed signs of HIV infection. What cells does HIV affect the most? And you already know it affects what? The cell immunity. Cell immunity. And we've talked about it that in cell immunity, we are dealing with what? T cells. But this one affects primarily the T helper cells. So we're talking about A. T helper cells. And sometimes I will say CD4 cells. So please take note. If you don't see CD4 cells and you see T helper cells, they all actually mean the same thing. All right. Preventive radioprotector was given to a worker of a nuclear power station. What mechanism from the below mentioned is considered to be the main mechanism of radio protection? Look at it. We've already analyzed that usually nuclear power plant or radiations are very, very problematic to the body, or they are very, it can damage the body cells. Okay. Now, they have provided what? A protection. So what is the mechanism of these protection, or how does it work? So basically, what they do is that they have, because in radiations, it inhibits what? Free ions, or they inhibit what? Free radicals. And that comes to destroy our cells. So in order to protect the person, we have to what? Inhibit these radicals. We have to inhibit these ions that are being produced from the nuclear power plant. Inhibit the radiations and you'll be fine. Inhibit the radiation. So here we are looking at for what? Inhibition of free radicals formation. All right. Now, X-ray examination revealed lungs emphysema in the patient. We talked about emphysema in our last class that we said that emphysema has to do with a gradual damage of the lung tissue. Specifically, it leads to thinning and destruction of the alveoli or the air sacs. And what happens in that case? When that happens, it means that you can breathe in, but you cannot recoil for the air to go out. So we have what? Hyperinflation, or we have overinflation in the lungs because too much air is accumulating, but they're not able to go out. So what is the term for that? We said there's what? Reduced elasticity or decreased lung elasticity. That's what? Inability of the lungs to recoil after it has what? Expanded. after it has expanded. Don't forget, when you breathe in, your lung expand. Then when you breathe out, it how do you call it? It shrinks or it recoils. Now in emphysema, that thing is impeded or it's not happening. An experiment on a dog, the peripheral part of the nervous vagus of the neck was irritated. What changes of the heart function would be observed? An experiment, a minute, guys. Okay. So an experiment, the vagus nerve was irritated. That means it was stimulated. What changes of heart function will be seen? Will be seen. So over here, we are looking for what? For the parasympathetic innervation. Parasympathetic innervation, because what? Vagus nerve. Vagus nerve always causes opposite in sympathetic nerve. Opposite in sympathetic what? Nerve. So in parasympathetic innervation of the heart, normally it is mediated by the vagus nerve. Mediated by the vagus nerve. And if I'm correct, this is the 10th cranial nerve. If I'm correct. But I stand to be corrected. The 10th cranial nerve. So specifically, the vagus nerve acts to lower the heart rate and hence decrease the contraction rate. So it will lower your heart rate. That's what the vagus nerve do. It will lower your heart rate. Vagus nerve to lower your heart rate. So here we are looking at what? Decreased contraction rate, because when the heart rate is lowered, that means contraction will be decreased. Contraction will be decreased. So your answer is D. Power input of a boy increased from 500 to 2,000 kilojoules per hour. What is the cause of it? Of course, if your energy is increasing or your energy input, your power input is increasing, what is the cause? Of course, you are doing what? Some serious exercise. Okay. And actually, everything here can actually lead to what? Increase in what? In power. Or increase in power. Everything here. Because if you're eating food, you use energy. Temperature, energy. Mental activity, energy. Physical, everything might use what? Energy. So all the options here can easily cause energy level to increase. But physical exercise requires the highest level of energy input. Requires the highest energy input. So here we are looking at for D. For D. All right. We have a student is thoroughly summarizing a lecture, okay? When his group mates begin talking, the quality of the summarizing worsens greatly. What type of inhibition of the cerebral cortex is the cause of it? It's just like when Sarah is trying to study, okay? So while studying, Paul and AJ and Lucy comes there talking about one nice movie they watched. Eventually, she might lose concentration. Because she might find that movie also what? Interesting. So might want to listen. Even if on a normal day she didn't prepare to listen to that, her focus was on to study. But now they are talking. So she's also listening. So this is what? An inhibition. But we call it what? External what? Inhibition. External what? Inhibition. And sometimes too, whether when you are learning on your own, you start thinking wide, like your mind is somewhere totally. That one's also there. That was internal, right? Because you are thinking, you are reading, but your mind is not there. Maybe you are thinking about, that summer holiday that you went to UK to spend the time with your friends and the swimming you guys went to. So that one is there. That was another part of inhibition. But this one is what? External. Because Paul and AJ are causing her to lose concentration. So here we are looking at what? External. And actually this phenomenon is called classical conditioning. Classical conditioning, which refers to a learning procedure in which a potent stimulus is paired with a previous neutral stimulus. I don't want to go into deep about the classical conditioning, but you can write it down in your own time, try and then go through it. Classical conditioning. Classical conditioning. So when a potent stimulus is being paired with a previous neutral stimulus. Previous neutral stimulus. Okay. All right. All right. So your answer is E. In the experiment of the animal, the part of the cerebral cortex hemisphere was removed. So your cerebrum was removed. It caused elimination of previously formed conditioned reflexes to light. To light. To light. What part of the cortex was removed? What part of the cortex was removed? Great. So we're talking about light. So your light perception are found where? They are found at the occipital lobe. They are found at the occipital lobe. Don't forget, temporal lobe is for hearing. So if the person had lost hearing abilities, we could be thinking of what? The temporal lobe. Then in post central gyrus, we are dealing with what? Sensation to touch. To touch. To touch. So in case you touch AJ and then she doesn't feel anything, please take her to the hospital. Maybe her post central gyrus is affected. They also have what? Pre-central gyrus. This deals with motor information. Motor information. And again, occipital lobe deals with what? Seeing or light stimulus. Seeing or light stimulus. That's why when somebody falls on her back, when the occipital part falls down or is injured, sometimes you can feel pain in your eyes. That should tell you that your eyes are actually at your back or at the back of your head. Now, the cerebellum deals with what? Coordination and movement. It deals with what? Coordination of movement and posture adjustment. Posture adjustment and coordination. That is what they do. So basically, that is just to talk about everything for you because you might meet them again. So I've taken my time to explain. All right. So your answer here is what? It's occipital. Occipital. We have inhibition of alpha motor neuron of the extensor muscles was noticed after stimulation of alpha of the what? Flexor muscles during experiment on the spinal cord. What type of inhibition can this process cause? What type of inhibition can this process cause? And this is called reciprocal. 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 And reciprocal inhibition describes the process of muscles on one side of a joint relaxing to accommodate the contraction on the other side of that joint. I repeat, it deals with what? The process or it describes the process of muscles on one side of a joint relaxing to accommodate contraction of or on the other side of that joint. It's just like you or those who lift, okay? When you are lifting, sometimes your bicep will relax so that your tricep will contract or will extend, okay? And again, your tricep will relax so that your bicep will contract. So that is called what? Reciprocal inhibition. That method is called what? Reciprocal inhibition. When one is flexing, the other one is what? Is extension. When one is extending, the other one what? Is flexing. That is what it means. So here we are dealing with what? Reciprocal. Reciprocal. All right. Respiratory coefficient was studied in the patient who strictly kept certain diet for 10 days. It was determined that it is 1. What diet was the patient following? what diet was the patient following? So first of all, you must know how to calculate respiratory coefficient. And respiratory coefficient, which is what KR, is equal to the volume of carbon dioxide produced. So volume of what? CO2 produced all over oxygen consumed, or volume of oxygen consumed. Volume of oxygen consumed, that's O2. Volume of oxygen consumed. So that means that in this particular patient, we're having volume of carbon dioxide and volume of how do you call it? Of oxygen. The ratio is what? Is 1. That means that we have what? 1,000 over 1,000. 1,000 over 1,000. To give us what? To give us that coefficient of what? Of 1. Of 1. And if it is 1, we are referring to carbohydrate. If it is 1, we are referring to what? Carbohydrate. If it is 0.7, we are referring to fats. Fat. If it is 0.8, we are referring to what? Proteins. Again, 1 is carbohydrate, 0.7 fat, 0.8 protein. 0.8 protein. So here, since it is since it is 1, we are looking at what? Carbohydrate. Carbohydrate. So your answer is B. Carbohydrate. Again, respiratory coefficient of 1 means what? Carbohydrate, 0.7 fat, 0.8 proteins. As simple as that. On the blood grouping on the system ABO, the standard serum of 1 and 2 groups cause erythrocyte agglutination. And serum group of blood group 3 didn't. What agglutinogens are in the erythrocyte? Now, when we talk about blood group O, okay. Now, I believe you know that A can donate to A, B can donate to B, and all of these things. So let's quickly go through it, and then we'll know what these agglutinogens are referred to. Okay. So what do we have here? So a blood group O is simply what? I. Okay. So I is O. I means what? So this I is what? Means O. I means A. Or that's blood group A. I is blood group B. IV is what? Blood group what? A, B. A, B. Okay. So we have, so now they said there was agglutination of what? Of I, that's blood group O, and blood group what? Blood group A. There was agglutination. Now, agglutination means that they did not what? Pick. They did not what? Pick each other. They didn't pick each other. But before that, let me tell you their antigens, okay? I will tell you their what? Antigens. So in blood group O, how do I put it safe? So blood group O has no antigen. Blood group O has what? No antigen. There's no antigen. No antigen. No antigen. So therefore, you will see no agglutination in blood group O. That is why everybody can receive from what? Blood group O people. Everybody can receive blood from blood group A people. Great. What about blood group A? Now, blood group A has A antigen. So let's use small a to represent the antigen. It has A antigen. It has A antigen. And therefore, it will agglutinate with blood group B and blood group O. Okay. So this blood group will agglutinate, or it will not match if you give somebody with O or somebody with what? With B. They will what? Agglutinate. They will agglutinate. So please You don't give. You don't give somebody with A and then you give somebody because that's antigen present. So it will agglutinate with B and blood group O. Now in blood group B, it has the B antigen. So let's say B antigen. It has B antigen. So what happens? It means it will agglutinate with A and O. So it will not accept anything from A and anything from what? From O. Now blood group AB is what? It has, how do you call it? Antigen A and B. So we have A and then what? B. So it will agglutinate with all blood groups. It will agglutinate with all blood groups. So that means that you can't give any of these things to somebody with what? With blood group what? AB. Sorry, what I'm saying is that you cannot give somebody, if you have blood group AB, you cannot donate for anybody. But you can only receive from people. You can't donate to anybody. So if that's what we call it, a universal what? Recipient. Blood group AB are called what? Universal what? Recipient. They can only receive, but they cannot give to anybody unless they are blood group what? Type. Unless they are blood group what? Type. And over here, they said there is agglutination of blood group A, blood group O, and A. There's agglutination, but there's no agglutination on blood group 3. No agglutination in the B group. So what agglutinogens are in the erythrocyte? And I said blood group B contains what? Antigen B. They contain antigen B. So here we are looking at for what? For antigen what? B. So your answer is what? A. Your answer is A. I believe I've taken my time to explain this. So if you don't understand, just let me know and I'll quickly go over it again when we are done. All right. So there's punctata hemorrhage was found out in the patient after application of a tourniquet with dysfunction of what blood cells is connected. So we're talking about a punctate hemorrhage. It's a punctate hemorrhage. This is a capillary hemorrhage into the skin that forms petechiae. It forms petechiae. So we're talking about a capillary hemorrhages or bleeding into the skin, into the what? The skin. And basically, what controls it is or are the platelets. The platelet. So over here, we are looking at what? At platelet. We are looking at what? At the platelet. We are looking at what? At the platelet. Because when platelets don't aggregate at the place of injury, what happens is that it cannot do anything. So coagulation cannot what? Take place. Coagulation cannot what? Take place. And of course, don't forget, we are talking about hemorrhage. And every other thing here has got nothing to do with hemorrhagic arrest. Okay, so here still we are looking at for it as for B, to clot. All right, but the rest are all inflammatory products or inflammatory cells. The rest are all inflammatory cells, but B is a coagulating what? Product. So platelets which are needed for what? To arrest bleeding, they are not there, or they are being damaged. All right. Student was taking examination, often have dry mouth, just like some of you were having in your Krok 1. They said this mechanism caused this state is the realization of the following reflexes. What I'm trying to say is that when you're taking an exams and your mouth is dry, what kind of reflexes is taking place? What kind of reflex is taking place? Don't forget that in exams, you are very, very, very conscious, isn't it? You are concentrating on your exams. So whatever is happening around you, you are not even what? Hearing. And sometimes you are filled with what? With fear. And fear and all those kind of, or anxiety, let me put it that way. Let me say anxiety. So if you have that kind of anxiety or having dry mouth, what comes to mind is what is a sympathetic system stimulation. Okay. And since you are focused on whatever you are trying to do, you are being what conditioned. You are being what conditioned. And so therefore here we can boldly conclude that this person is having what conditioned sympathetic reflexes, conditioned sympathetic reflexes. Now, in unconditioned, we are referring to what? To something that you are not what learning, or an unlearned reaction to stimulus, and unlearned reaction to stimulus. In other words, it is not planned. You don't plan it in unconditioned. You don't plan it. It just happened. That is unconditioned if you don't plan it. But if you plan it, or you are consciously focused on something, it is called conditioned. Conditioned. Like I said, presence of dry mouth, anxiety, all of those things are all signifying what? A sympathetic effect. So your answer here is conditioned sympathetic. All right. The middle part of the cochlea of the internal ear was destroyed in an animal. Middle part. It causes abnormalities of the sound perception of the following frequencies. Now take note of this, because this question likes coming a lot. So first of all, you must know that the organ of corti is the receptor organ for hearing. Again, the organ of corti, it is the receptor for hearing, and it is located in the cochlea. It is located in the cochlea. Now, for high frequency sound, you can find it at the base of the cochlea. So write high frequency base of the cochlea. Great. That is, it is near the round or the oval window. Round or oval window. If you have done anatomy of the ear, you will know what the round or oval window is all about. So for high frequency, we are talking about the base. The base. For low frequency, we are looking at what? The helicotrema or the apex. We are looking at what? For the apex or near the apex. Also known as what? Helicotrema. Please, I'm taking my time because you're going to see questions on it plenty times. Helicotrema. Then MA. Helicotrema for low, for high at the base or the oval window. Then, of course, in between, we have what? The middle. So for middle frequency, you can find out what? At the middle part of the helix. For middle, you find out what? At the middle part. So just as the name implies, middle. So I repeat, high, go for base. Low, go for the apex or helicotrema. And you will see these words most of the times. So again, since it's middle, we said middle means what? Middle. So middle will be the answer. So in high, you could be hearing for base. In low, apex. That's just by the way. The person has decreased diuresis. That means the person is not urinating. Hypernatremia. That's to tell you that there's sodium retention in the body. And we know that sodium is controlled by what? Of course, by the adrenal gland. To be specific, the aldosterone. When aldosterone is secreted, it retains what? Sodium to prevent urination or to prevent diuresis or diuresis. There's what? Hypokalemia. And you know that if there's aldosterone level are high, definitely kalemia will move in the opposite direction. That's why they are being secreted, and hence hypokalemia. So what hormone is being secreted? Again, we are dealing with what? With aldosterone. So the answer is D, aldosterone. The temperature of an ambient environment is 38 degrees Celsius and relative humidity is 50. What ways of heat emission provide maintaining a constant temperature of the human body? maintaining a constant temperature of the human body. Now look, temperature is 38. That means it is quite hot. Humidity is what? Is 50. It's 50. It's 50. So that means that if you are hot, what happens is that fluid on your skin or the sweat on the skin will begin to what? To evaporate, to cool you down, right? It will evaporate to cool you down. So this process is called what? Evaporation. And evaporation is a way that the body dispatches heat to the environment via sweat, via sweat, via sweat, via sweat. And anytime you see relative humidity, know that they are talking about what? About fluid or sweat that's about what? To leave. And that is your clue too for that. And of course, the temperature can also help. The fact that it's very hot on the day, so you will be sweating. And if you are sweating, definitely you will lose water or heat will be lost. Heat will be lost. So here we are looking for what? For evaporation. Another question, I'll show you the difference between the radiation and the conduction and all those stuff. So don't worry about that. But for now, understand that when you're having a humidity of 50% or just humidity and high temperature, you are looking for what? For evaporation from the person or evaporation of lakes or water bodies, basically. Okay. So basically, that is it.